So my name is Gábor Varga, and I will give this seminar lecture today. And since, as most of you know, that I am actually a basic scientist, I mean, my background is biology. So I will tell you some of my, I would say that, actually two stories. One is from my past gastroenterology or gastrointestinal physiology background, and another one is from dentistry. Here is the main message. Then you can actually leave, maybe. I mean, here there is a list of people. Professor Silvester Vizi, who is the president of the Hungarian Academy of Sciences. Professor Carmelo Scarpignato from University of Parma. I mean, they are both actually pharmacologists. And then we worked together. I mean, they were actually more prominent than me when I was young. Okay, next one is Maynard Case, Professor Maynard Case from University of Manchester, who is an absolutely great physiologist. Also, he was the dean twice at the Biomedical School of University of Manchester. And his research was in, he's still alive, but he retired from science on the pancreas and salivary glands. Next one is Martin Stewart, who actually worked together with Maynard at those times. Even today, that he shows up at Semmelweis as our visiting professor, and actually we work together with some basic science projects. But the lower line is that Professor Bruce Baum from NIH, not only a salivary researcher, but an absolutely great organizer and figure actually in dental research. He has also retired. Next one is Professor Pamela Dambestan. She has not been retired. She is at University of California, San Francisco. She is a great researcher in the field of enamel research, actually, one of the top researchers in the world in enamel research. The next one, actually, a guy that from the Netherlands who worked together with Pam, and then he connected us, Tom Bronkers, who connected us with Pam, and actually, as a result, actually, we have two joint grants from the NIH, NIDCR, so from the American National Institute of Health, which was a great honor, I have to say, and then we also actually contributed quite a bit together to that field. And the last one is in a younger format, Peter Hegyi, a pancreatologist, I would say. Originally a basic scientist, today translational medicine, research, whatever, I mean, many things together. What is common in these guys, they are almost great scientists. And also, actually, they are also very good organizers. I mean, at different level, of course, but I mean, Vizi, Dambestan, Bruce Baum, Peter Hegyi, they are certainly top administrators organizing things too, I mean, at world level, actually. And then I had the chance to work together with such guys. So the message is, please try to work with people like them. What is common in them is that they are not only great scientists and great organizers, but they are also good persons, in my eyes. Fair persons. That's very important in science, to work with people that are actually not very good, but also very fair. All right, so this was the major message, and now comes my two adventures. Since I graduated in 1980, I did many different things in my life, I mean, for research. But there are two examples which may give you actually some clue about my, I would say, the destiny, something like that, and also give some perspectives, because in both, one is that from the gastrointestinal physiology and pharmacology, and this one the first, which I actually did between 1980 and 2010, and another one is salivary research. which started in 1990-ish, and then still going on. All right, so cholecystokinin. If you do teaching in physiology, or you remember your physiology studies, I mean, you must know about this hormone and neurotransmitter. Originally, it was discovered as a peptide, which actually contracts the gallbladder, but later on it was also recognized that that is very important. That has important functions in many GI things, particularly in pancreatic secretion, and also actually gallbladder contractions, gastric emptying, and bowel motility, but also actually has an important role in controlling satiety. So, am I going to be fat or thin, kind of things. And then, of course, as I came from the biological side, so my original studies were biological, and then but from the beginning, I tried to do and get involved in things which is useful, I mean, which could be useful. And I have to say that these first slides, I will not explain all of them. Still, I put quite a few slides, so you see that I'm actually in a 30 years kind of time frame. I and with the people that whom I worked together, I mean, we did a lot. But at the end, actually, this is a kind of failure, at least in terms of having something, a pill or medicine, which is given to patients now. And I will also show you why. So again, these multiple roles, I mean, CCK is actually released not only from intestinal special cells, which actually senses the food, some food components, and if those are there, the pancreas is stimulated. I mean, that's the hormonal regulation, but it also acts as a neurotransmitter. I mean, there are CCK-ergic receptors all around, and most of the motility stuff is actually related to the CCK as a neurotransmitter. And also, I mean, as we will see, that the food intake control. What is interesting on this, I mean, that CCK, or cholecystokinin, is very similar to gastrin. Actually, the major difference is the sulfation and the 7th position in the active part of the molecule, and then makes the big difference, as gastrin is primarily for gastric acid secretion, and CCK is primarily for this kind of pancreas and motility stuff. Of course, because there are two receptors. The CCK1 receptor is sensitive to CCK only, and not to gastrin, while the gastrin receptor is sensitive to both. But there are some other kind of fine-tuning on this, on these two. So not all CCK1 receptors are born equally. So actually, there is a number of CCK receptor antagonists, CCK gastrin receptor antagonists. We call them CCK1 and CCK2. The second one is CCKB, or gastrin receptor, to make it more confusing. But anyway, so we will look at actually examples for those, because again, I'm coming not only from physiological perspective, but also in GI pharmacology, working together with people like Visi and Scarpignato. As we look at the potential therapeutic targets, I mean, where to use any kind of CCK agonist and antagonist, of course, those are the places when the receptors are there. So the pancreatic acini, the GI, smooth muscle, pancreatic ducts, and also cancer cells, actually, in the pancreas. And what is in some ways is very interesting, that the difference of CCK1 receptor activation in different targets. I mean, you know that this smooth muscle contraction, I mean, in the gallbladder. So if we look at this, wait, I have to move this. Okay, this second red line. I mean, as you see that a kind of maximal stimulation on contraction happens only in very high doses. So high, I mean, of course, I mean, it is high. And then the pancreas, which is the continuous red line, I mean, is very sensitive to CCK to up to a point of 10 to minus 10 mole. But when you increase the concentration of CCK, you get a decrease in pancreatic amylase release. And this is not only a decrease in the release, but that is also a kind of phenomenon when the pancreas becomes inflamed. This is in those who work with pancreatitis, this is the so-called CCK or sertraline-induced. Sertraline is an analog of CCK-induced acute pancreatitis. So there is this biphasic effect in the pancreas, and there is a monophasic and shifted to the right effect on the muscle. So although the sequence of the CCK receptors, these are all CCK1 receptors, but the response for activation is quite different. Also, there is a special analog of CCK, a partial agonist, which activates only the muscle. It activates only the increasing phase, both in the pancreas and in the intestine, but not the decreasing. So this GMV, this compound, CCK analog, it doesn't cause pancreatitis. If we go to the receptor antagonist, since there has been lots of possible targets, so there were quite a few actually activities from pharmacological companies to develop antagonists for those, to inhibit CCK-like effects. And if we look at the structure of these, the advantage of such a structure is that these are not peptides. You see, when you use peptide analogs, that breaks down very fast, just like the peptide itself, but these are actually analogs which are other kind of structures, so therefore stand very well against, absolutely actually against proteolytic enzymes and things like that. Now, I just have a couple of slides. I don't want to bore you with the pharmacology of this. In these studies, actually, through many years, actually, that we studied different CCK receptor antagonists, and then we described that how they behave in gastric emptying, in the rat, or in human gallbladder contractions, and then how can those be inhibited. And we also used actually isolated guinea pig pancreatic ducts. I would not go to the details on this. This is a very special technology, actually isolating these little ducts, and then you can look at the function by measuring actually the change in intracellular pH as a measure of bicarbonate secretion of these cells. The right side is some kind of dose dependence. This one too. And then we also did some further studies, actually look at the satiety effects of CCK agonists and antagonists. So satiety means that if you inject it, the animals either eat less or more. Okay? And then we did the approach in two different ways. One was that we used monoclonal antibodies, which are in the circulation, so that could neutralize the endocrine function of CCK, while the specific antagonists, which are non-peptide-like things, they just penetrate everywhere. They can act on neuronal kind of nerve endings too. So this slide just clearly shows that when you use this, and actually this slide and the next slide was about the food intake. You see, today many people take this kind of drugs like GLP-1 agonists. As you know, I mean, this is very famous now. Many movie stars, many kind of people who would like to look beautiful, I mean, take these kind of things. Those are very effective, actually, and so these kind of GLP-1 agonists. So at that time, when I was a young scientist, that we were hoping to have kind of similar effects with CCK, and in some ways it happens, in some ways not. And then there was a kind of this almost 30 years lag period until a real effective, selective, whatever, this kind of GLP-1 agonist again appeared in the market. So now this slide just only shows that this is pancreatic amylase secretion, and that shows that if we use monoclonal antibodies, then with the monoclonal antibodies, we can actually inhibit the CCK-induced secretion. So if we give the circulation CCK and then monoclonal antibodies, then we can neutralize the CCK effect. Now, which had a great promise, but also the disappointment in some ways. Now, when we do the same thing for food intake, then monoclonal antibodies had no effect. I mean, that's on the left. Okay, left. Right here. Okay, this is the cumulated food intake, and this is quite physiological because what we did is that we used in a dark room. I mean, we measured the food intake of rats when we sleep, and then they wake up. Because when you turn off the light, and in animal houses, that's the standard way, 12 hours light, 12 hours dark. Rats eat 95% of the food when the light turned off. Okay, so that's the spontaneous normal food intake. So this is the period when you could really study different kind of compounds, whether they affect the food intake, increase or decrease. Now, what we saw here that the monoclonal antibodies had no effect. Okay. But we also saw that when we use the CCK receptor blockade, and this time actually we combined it and blocked both CCK A and B receptors, the animals R by R actually ate more, just simply getting CCK receptor antagonist. Now, I don't have a slide on that, but if you give CCK itself, you can actually inhibit the food intake. So that could be a very good satiety hormone, except that what I already showed in the other slide, and this actually blocked this project, is that when you activate pancreatic secretion by CCK, then over a certain dose, I mean, this kind of so-called supramaximal doses, I mean, destroy the pancreas. And then the motility change, which actually is the mechanism for food intake control, not the only one, but an important part, that is just activated over that dose. So how can we live with this? Of course, because locally at the synapses, you see, I mean, CCK concentration could be very high, but not in the circulation, not in the way what reaches the pancreas. So the bottom line is that since the concentrations of CCK, which actually cause satiety effect, also destroy the pancreas, so therefore we were unable to use this as a treatment for humans to decrease food intake. That's quite different from GLP-1 agonists, because if they do anything in the pancreas, that's an inhibition of pancreatic secretion, but we don't have this kind of biphasic and devastating effect. There was still space for applications. Irritable bowel syndrome, you see, this is something that either constipation or the opposite, some kind of altered defecation. And this is quite common actually in the society, in the population. And then, since CCK has a strong motility effects, so that was along with many other hormones. So we put it up, and actually this was an international network that here we did kind of in vitro and in vivo motility studies. But we also worked together with clinicians. I was involved in clinical trials too on this way, that we wanted to test whether CCK antagonist in this way could be a good kind of compound, actually, or good compounds to affect this. And we put it up because that has been known at that time that actually CCK1 receptors are there. And then the primary effect is this kind of neurotransmitter effect, and primarily on nerve. The action is on nerve endings, and then secondarily on the muscle. So, and indeed, I mean, just to take it quite short, that when you look in normal humans, so in non-diseased humans, in normal humans, when you look at the proximal colon propulsive motility, and then what we clearly saw is a strong inhibition. Okay, a strong inhibition of this motility, which actually is against the normal kind of colonic activity. So now we also looked at what happens with the colonic transit. And as it appears that in those who have actually a delayed colonic transit, so the content of the intestine slows down, that's the delay, then actually it could be put back to the right track when you use this CCK antagonist. So this was quite promising. And also there were actually two studies, just to make it short. I mean, these are many years actually, what's happening. And then this was led by an Italian company, Rota, who actually developed these antagonists. There was a clear evidence that this CCK antagonist, dextroxyglumide, actually alleviates the IBS symptoms in one phase 2 study, and then some other studies also confirmed this. So at that time, I felt that I did something really useful. And then one day, I mean, there were many other options besides CCK, and today many others are used. Well, what unfortunately happened that even the drug was effective, but the company stopped because they moved to another area, and then maybe because the effect in humans, you see, I mean, especially in diseased humans, I mean, because of the personal differences in some patients, there's a strong effect, in other patients no, so finally they decided to stop. So many of the activities what I did in my first, I would say the 30 years research, this was just a part of that, but this is particularly about CCK, didn't lead to a drug development. Okay, it is still there. Maybe one day it comes back. We will see. But right now you cannot buy a CCK antagonist to treat IBS. My other adventure, which was a little more luckier, although I have to say that I didn't get rich of this. So this is not about money, but this is about science. And again, I'm a basic scientist, and I have been doing basic science for quite a while. But whenever there was a possibility to translate that knowledge to clinical applications, I either therapy or diagnostics, that I at least I tried. This is the digestive tract. So, and actually, gradually from a research institute, I mean, I worked for the Institute of Experimental Medicine for 22 years, but gradually I moved to the university, and then they invited me here to the to the Department of Oral Biology to lead it. And the reason for this, because although originally I studied the pancreas and other parts of the GI tract, but the salivary glands in many ways are quite similar to the pancreas, so many of the research is actually overlapping. I would just jump through on this, and instead, I show you the two. Salivary research is not the #1 research area of the world, I have to say. But there were two periods, so there were two Nobel laureates actually who got research because of salivary gland research too. But it wasn't only salivary, because one of these was Pavlov, who did his studies in the late 19th and early 20th century, who worked in Russia. And then he is actually the founder of not only GI and oral physiology, but also neuroscience, as it is, I have to say. So, but for long, nobody knew about his work because he worked in Russia and then he published in Russian. Until 1902, when he was invited to the Royal Society to give a series of lectures. And then his research attracted so much, those guys who were there, so they decided that his work should be translated, which was already written in Russian. The work of the digestive glands, I don't know the Russian names, although at certain points I knew Russian, but I couldn't translate it back. Anyway, so this was translated to English. That became very popular, actually, for those who are interested in health sciences. And then he got the Nobel Prize in 1904. The other one, and we have a debate with Peter Hegyi, I have to say, he and me, because he's a pancreatologist by heart and I am a salivary researcher by heart, that why George Pallad received the Nobel Prize, because of his salivary research or pancreas research, and of course for both. Because what he did is that he was the one who actually discovered exocytosis, but not only exocytosis. As you know, endocrine and most endocrine and practically all endocrine organs, produce their product by stimulation. If that is a protein product that by stored proteins in zymogen granules or some kind of other granules within the cells, why the synthesis continues. So of course, the peptide is synthesized in the endoplasmic reticulum, and then it is transported to the Golgi. Secondary changes happens in the Golgi, and then there are some condensation in these vacuoles, and then the cells store them. So this process is continuous, although regulated, but continuously happens. While the secretion happens only for certain stimulation, like Sunday dinner. We see the food. And those who start to eat. Lots of saliva is coming, and then in the secondary phase, the stomach is activated, and then pepsin is secreted, and then in the third phase is the intestinal phase when the pancreas and the pancreas secretes and the gallbladder contracts. So actually, he figured out the molecular mechanism of this. And then this is one of his major papers, which is actually on the parotid. But indeed, Peter is right that in his Nobel lecture that he first mentioned the pancreas and then only second the salivary glands. Anyway, the lesson here is that if you have a certain kind of process, certain kind of disease, just look around to similar organs, similar diseases, or maybe different diseases, because that gives you a very good idea that why it may give you a good idea that why certain disease happens in one organ, it doesn't happen in another one, and then you can compare the the common things and the differences, that could be quite fruitful. So this is the pancreas. This is the salivary glands. Okay. Why to study the salivary glands? I mean, the salivary glands, there are multiple diseases, but then there is one prominent one, and this is Sjögren's syndrome, which is an autoimmune disease, one-way street, slowly develops, but it could take away the full salivary gland gradually. I mean, so the SNR part is lost. Today, we do not have an adequate treatment to reconstruct this. So actually, what we did here is, again, some kind of really fundamental science, mainly what we did, is that we tried to do two things. One is to isolate cells from human, actually, subendomal glands that we got from the oral surgery clinic. And then, on one way that we did some tissue engineering methods to reorganize and redifferentiate those cells which were isolated. And another way was that we studied actually the molecular mechanisms of ion secretion in those, just to model that how it happens. So this is not a direct lead to a treatment. So this is a kind of basic science. What I said, I mean, I described this morning. I mean, just very shortly going through, I mean, this is as it looks like, that we isolated. I mean, the individual cells could actually divide, and then they can start to reform if you use the right, I mean, reform a kind of acidotubular structure if you cultivate them in the right conditions. So this is just some demonstrations of those that I don't have time today to describe in details. And then we also use these cells, you see, epithelial cells, that applies to the pancreas too. If any kind of epithelial cells, if you can manage them to survive, what they do, that you put them on the surface, they form a monolayer. They develop tight junctions, okay, and then any kind of thing which goes through this epithelial surface, I mean, it is actually controlled by the cell. So ions are moving through, actually, especially kind of salivary cells and pancreas cells. The leading ion is chloride, as we know, I mean, in the SNI, and then that is followed by sodium paracellularly, and then water through the cells. But it has not been known, I mean, the details of the molecular mechanism. And I have to say that, I mean, this is one of the methods, actually, what we used. I mean, this is a fairly old, but still preferred method to use, actually, to measure, I mean, to build up a monolayer like this. You can also do that in intestine, actually put that in kind of chamber, like to think, and measure, just simply measure the ion currents. Because the ion currents could show the level, and then just measuring actually the movement of electrons, you see. I mean, the ion current is a good measure of epithelial function. And then you can play around with this, I mean, using different agonists and antagonists to follow the procedure. By the way, that Peter Hegyi and I also did actually in the past this kind of research from the pancreas too, and then we utilized that in the salivary research too. I mean, again with these human cells, and I will not explain the details. I just show you that we generated quite a bit of data. And then we tried to push the cell, we tried to push the cells to redifferentiate to SNR type cells from the non-differentiated ductal types. But this is very fundamental basic science. So again, as a basic scientist, for long I thought that I would never really go to a clinical application, a potential clinical application of this. And then when I saw this paper, I said that, well, this is really interesting. When these two guys actually published the paper in the Journal of Dental Research, which is the number one, number two, I mean, top journal in the world of dentistry. And then what they published besides classical salivary stuff is that there is a high potential in saliva to measure exosomal microRNAs. I mean, I assume that everybody knows what exosome. I mean, this little kind of membrane-coated particles which actually secrete microRNAs of the cells which the cells produce. And the beauty of this is that these microRNAs not only tissue-specific, but also disease-specific. So, for example, for oral cancer, so we are still at the proof of concept. But for oral cancer, if we can isolate microRNAs from saliva, and then we can multiply this by PCR, okay, then actually we are able to identify a very early stage of an oral cancer. But what these guys did is that they also went after some other cancers. And really, I mean, the development of this area is really very slow. But what they assumed, and then they provided some evidence for this, I mean, these microRNAs which appear in the circulation, for example, for pancreatic cancer or gastric cancer or pulmonary cancers, not only kind of go to the circulation, but the salivary glands, which are over there, okay, on the right, pick up these exosomes, and in a transcellular process, actually, they transport them to the saliva. If this is true, this could be a fantastic technology to continuously screen people. For example, in an elderly home, when there is 100 patients, I mean, and then this, not patients, I mean, old men or women. I mean, sooner or later, they will get, at least half of them will get cancer. Okay, that would be good to identify that early. But you cannot take blood in every week. But they can spit, at least most of them. I mean, every week or even every day. So if, again, this, right now, this is at the proof of concept level, but if this is really a reliable, if the reliable and sensitive specific method could be developed, then this could be a very good way to actually to screen lots of people who otherwise will not, and also, I mean, that could be quite cheap, inexpensive. All right. So I said, yes, this would be great, but we would never do this in Hungary. But then SARS-CoV-2, I mean, came not only to China and to other parts of Europe, but to Hungary too. Again, I don't have time to detail. This is an RNA virus which are produced by the cells, and actually primarily, at least the first, it affects actually the airways. So therefore, that's fairly easy to measure them if you do a polymerase chain reaction. If you collect nasopharyngeal samples. And this must be, if the method is correct, is set, it must be a very sensitive method and more sensitive. I mean, well, proteomics are fine. But this must be much, much more sensitive, because if you run 25 cycles, I mean that you will get 33 million copies, which is, in many cases, still acceptable if you have good primers. Even the 35 cycle brings specific and sensitive results. That's 34.4 billion copies. So the classical way was this nasopharyngeal sampling. I hated that. Probably those who actually did the connection too. So at the very early, actually, at the time when we saw this, at the very early stage, that we thought that we have to do something. And since me and my fellows actually were already involved in translational medicine, actually, that we kept going to page. I mean, Mark Zumba was one of those of the 20 or something that I showed in the morning. And actually others also, I mean, that from the dental school actually went there, since we had a kind of joint project with Peter from Semmelweis. So we thought that, I mean, saliva should be good enough too, I mean, to measure the samples, but it had to be kind of proved. And this was not easy with the clinicians, if you look at the clinical side, because in the mind of the laboratory medicine people, saliva is very sticky, very hard to handle, very difficult to do anything with those, so they hated that, okay? The first thing what we did is that we wanted to test it by the available data, actually not surprisingly, with the meta-analysis. So the COVID breakout in Europe in March, mid-March, in Italy a little earlier, maybe February, but it reached actually Hungary in mid-March. We published a paper on the available weak evidence. So there was just a relatively small number of small sample size studies, but that was already enough, since we had this kind of methodology, I mean, meta-analysis engine at that time, with the statisticians, with the way of thinking. And so already in August 4, in 2020, we published a paper in Frontiers of Medicine saying that salivary samples are at least as good as nasopharyngeal samples. Sorry about some Hungarian words on this slide. Actually, this was, I mean, the person who was the first author on this was Mark Zumba, that many of you actually already know that he is now a specialist at periodontology. Actually, he was here in the previous session. And Anna Földes is a basic scientist who is my colleague at the Department of Oral Biology. And actually, I mean, we are not Yale University. Still, I mean, we came up actually with a relatively strong evidence 1 month before them. But of course, I mean, they did vet science, and then we did meta-analysis, so maybe on one way this is not fair. But if you think about the level of evidence, I mean, that was, I mean, we provided actually a strong level of evidence. And at that time, that was very important. So by 2002, when we looked at what is in the journal, that they count the views and the downloads and the citations. So this was a very early time of citation, maybe 2020, and not 22. And there were already 23,000 kind of downloads and views. And then by now, when I looked at the website, that there were 44,000 views. I mean, citation maybe not that high. I mean, 250. Although this is one of my best papers quoted, and even for CTM, I mean, with this short period of time, that this is maybe in this 5-year period, this is the best quoted paper from here. But this is not only the numbers. But what happened here is that there was really an unmet need. And then was really a kind of hypothesis that we thought that this could be important, and then we were able to act immediately because we had the network, actually methodologically run by Peter Hegyi. And then we actually went farther on, and since the big problem is the rheology, I mean the stickiness of saliva, so we did quite a few studies actually here at some pharmacists, some top pharmacists from our university, and then we were able to figure out that some of the classical methods which affect viscosity, they don't work on saliva, just very simply freezing and thawing. We were surprised, but there was no change. I mean, it was still sticky. And then we warmed up to some levels, it was still sticky. But also, which was a very good coincidence, is that the cocktail which I used for nasopharyngeal samplings contained quite a few components, and one of them was proteinase K, which actually breaks down the stickiness, so the mucins. So, and then we could clearly show that actually, and then also or laboratory medicine people accepted that saliva could be handled quite well if you use this kind of cocktail. And as a consequence, I mean, we could do quite a bit of stuff. We also allied with the company, their name is Omixon, and then together, I mean, we did the testing on the samples, and then they did the development modification. So this is a kind of very good biotechnological company. And then finally we figured out that this is a very good method for doing the samplings. And what we also figured out as, I mean, this is a complicated figure, but let's say that the next COVID is coming. Okay, and then you have to test many, many people. I mean, there are multiple ways, right away. One way is that in a school or in a university group or in a department, I mean, you just pull them. Pull the samples, okay, and measure the individual samples. And then, in this way, each sample actually contains 40 samples from the patient, from patients or the people who are involved. And then you have to retest only those when you get positivity, and not the others. So this is this kind of pyramid-like method. I mean, it could be quite useful when the next COVID virus or next whatever virus comes, and then we can use that. And then we also, in many ways, I mean, with the company, with this biotech company, I mean, that was their product, but we did the biological side of that. I mean, now we have a kind of set arrangement that we not only did the meta-analysis, but then with Omixon, actually, we did the optimization of virus detection for salivary samples. And although it didn't make a big financial, not a big, even no financial, I would say that upgrade for either the company or us, but this is a very good addition to science. The other conclusion, I mean the failure side, is that by that time when we were ready with this optimized test, I mean, that still cost money. And the government, not only here, but in many other countries, because they were worried, you see, I mean, they bought up so many Chinese and whatever, I mean, tests, so they gave it out free. So a hospital director would not buy it for money if they can get it free. So therefore, there was no financial benefit, but there is a strong I mean, promise for the future that we can use this kind of methodology one day when, I mean, just little modifying the primers and so, I mean, for this. And one of the consequences is that Regina, my PhD fellow, I mean, that she actually started she's a brand new PhD student, that she started a meta-analysis actually, not on viruses now, but we want to use the knowledge what we had, and also that this kind of knowledge about exosomes to figure out whether head and neck cancer prognosis can be detected by exosomal biomarkers. And then hopefully, as a next step, actually, we can continue with salivary test, and we are already building up actually connections for clinical colleagues. So there is a promise here. Again, maybe this is the most important slide. Try to work with bright guys who are fair too. And then you could be very, very successful. And anyway, thank you for coming. Thank you for listening for me. I mean, I'm ready to answer questions. This was just a small fragment of my research in the last 46 years or so. I mean, lots of years. But I think that there were two kind of clear procedures. Some of them went to kind of at least relative failure. Another one, I think, is scientifically a big success. But altogether, I'm very proud of having, I mean, these guys kind of guiding me, and then working with me in parallel, and I wish you the same thing, actually, for your future. Thank you very much. Oh, by the way, I have one more slide. This is my grands, this is my family. Actually, only 4 grandchildren I show up because I'm very bad at collecting the pictures. For science, you need money. And then this is some of the projects, actually. Right now, I have funding sources from 2 different sides, but to me, what is very important is my wife, who has been supporting me for 48 years or something like that. I mean, more, but we married 48 years ago. And then those are on these young guys, my 4 children, and then the wife of the youngest one. And then I had to say that since I trust in God, and then we got so much undeserved help, me and my family, so soli Deo Gloria. Thank you.